Hello students, welcome to readmedprep academic channel. Today in Human Health and Diseases Part 4, we are going to talk about the basic concepts of immunology. In this, we are going to talk about the types of immunity, immune responses and the lymphoid organ. Now let us start about the immunity and the types of immunity. Immunology is a study of immune system. This system protects an individual from various infective agents. It refers to all the mechanisms used by the body for protection from environmental agents that are foreign to the body. When the immune system does not function efficiently in an individual, it leads to infection causing disease. The overall ability of the body to fight against the disease causing pathogen is called immunity. It is also called disease resistance. And the lack of immunity is known as susceptibility. Immunity is very specific. Normally, many of the responses of the immune system initiate the destruction and elimination of the invading organisms and any toxic molecules produced by them. These immune reactions are destructive in nature and are made in response only to molecules that are foreign to the host and not to those of the host itself. This ability to distinguish foreign molecules from self is another fundamental feature of the immune system. However, occasionally it fails to make its distinction and reacts destructively against the host's own molecules. Such autoimmune diseases can be fatal to the organism. Almost all the macromolecules, example proteins, polysaccharides, nucleic acids, etc., as long as they are foreign to the recipient organism, can induce an immune response. Any substance capable of eliciting an immune response is called an antigen. Expansion, antibody, the anti is taken, and generator, gen is taken. So, antigen. There are two broad classes of immunity responses, namely innate immunity and acquired immunity. Here you can see the classification. Immunity is of two types, innate immunity and adaptive immunity. Adaptive immunity is of two types, natural immunity and artificial immunity. Natural immunity is active because of infection or passive because of the maternal antibodies. Artificial because of active immunization or passive because of the antibody transfer. Innate immunity. It is the natural phenomenon of resistance to infection which an individual possesses right from the birth. The innate defense mechanisms are non-specific in the sense that they are effective against a wide range of potentially infectious agents. It is otherwise known as non-specific immunity or natural immunity. Here you can see a number of innate defense mechanisms are operative, non-specifically against a large number of microorganisms. In the image on the right side, you can see the various anatomical and the physiological barriers of microbiological attacks. In the tears, there is lysozyme, removal of airborne particles through the nasal mucosa. In the bronchi, there is mucus and cilia. In the gut, the hydrochloric acid is present. In the skin, it acts as a physical barrier. Rapid pH change occurs below the stomach, in the duodenum and the intestines, and the flushing of the urinary tract, and the low pH of the vagina. Now, in this table, we can see the type of innate immunity and the mechanism it follows. Anatomical barriers, the skin and the mucous membrane. Skin prevents the entry of microbes. Its acidic environment, pH 3 to 5, retards the growth of microbes. Mucous membrane, mucus entraps the foreign microorganisms and competes with the microbes for attachment. Physiological barriers, temperature, low pH, chemical mediators. Temperature, normal body temperature inhibits the growth of the pathogens. Fever also inhibits the growth of pathogens. Low pH, acidity of the gastric secretions, hydrochloric acid in the stomach kills most ingested microbes. Chemical mediators, lysozyme acts as an antibacterial agent and cleaves the bacterial cell wall. Interferon induces antiviral state in the uninfected cells. Complementary substances produced from the leukocytes lyse the pathogenic microbes or facilitates phagocytosis. Phagocytic barriers, specialized cells like the monocytes, neutrophils, tissue macrophages, phagocytose and digest the whole microorganisms. Inflammatory barriers. Tissue damage and infection induce leakage of vascular fluid containing chemotactic signals like serotonin, histamine, and prostaglandins. They influx the phagocytic cells into the affected area. This phenomenon of movement of the phagocytes to the site of inflammation is called diapedesis. Now let us move on to the acquired immunity. The immunity that an individual acquires after birth is known as acquired immunity. It is the body's resistance to a specific pathogen. The unique features of acquired immunity are antigenic specificity, diversity, recognition of self and non-self, and immunological memory. What are the components of acquired immunity? There are two components: cell-mediated immunity or CMI and the antibody-mediated immunity or humoral immunity. Cell-mediated immunity, when pathogens are destroyed by the cells without producing any antibodies, then it is known as cell-mediated immune response or cell-mediated immunity. This is brought about by the T lymphocytes or the T cells, macrophages, and the natural killer cells. Antibody-mediated immunity or humoral immunity, when pathogens are destroyed by the production of antibodies, then it is known as antibody-mediated or humoral immunity. This is brought about by the B lymphocytes or the B cells with the help of the antigen-presenting cells and the T helper cells. Antibody production is the characteristic feature of the vertebrates only. What are the differences between the humoral and the cell-mediated immunity? The main cells involved in the humoral immunity, B lymphocytes. In cell-mediated immunity, T lymphocytes. And the antibodies are involved in the production in humoral immunity. Whereas cell-mediated immunity, antibodies are not produced. And the pathogens are identified by the antigens that are floating in the blood. Whereas in cell-mediated immunity, by the antigens on the surface of the infected cells. The pathogens are killed by the antibodies, whereas in cell-mediated immunity by specialized killer T cells. Now, the cells divide into either plasma cells or memory cells in the case of humoral immunity, whereas in cell-mediated immunity, the cells divide into different types of specialized T cells, like the T helper cells and the T killer cells. What are types of acquired immunity? It is active immunity or passive immunity. The immunological resistance developed by the organisms through the production of antibodies in their body is called active immunity. Active immunity is acquired through the use of person's immune responses, which lead to the development of memory cells. Active immunity results from an infection or an immunization. Passive immunity, it does not require the body to produce antibodies to antigens. The antibodies are directly introduced from outside into the organism. Thus, passive immunity is acquired without the activation of a person's immune response, and therefore there is no memory in passive immunity. What are the differences between active and passive immunity? The active immunity is produced actively by the host immune system, whereas passive immunity is received passively and there is no active host participation. Active immunity is produced due to the contact with the pathogen or by its antigen, whereas passive immunity is produced due to the antibodies obtained from outside. Active immunity is durable and effective in protection, whereas passive immunity is transient and less effective. Active immunity, the immunological memory is present, whereas in passive immunity, there is no memory. Active immunity, the booster effect on subsequent dose is possible, whereas in passive immunity, subsequent dose is less effective. In active immunity, it is effective only after a short period, whereas in passive immunity it develops immediately. Now, let us move on to the immune response. The types of immune responses, the primary immune response and secondary immune response. The primary immune response occurs when a pathogen comes in contact with the immune system for the first time. During this, the immune system has to learn to recognize the antigen, produce antibody against it, and eventually produce memory lymphocytes. The primary immune response is slow and short-lived. Now the secondary immune response, it occurs when a person is exposed to the same antigen again. During this time, immunological memory has been established and the immune system can start producing antibodies immediately. Within hours after recognition of the antigen, a new army of plasma cells are generated. Within 2 to 3 days, the antibody concentration in the blood rises steeply to reach much higher level than the primary response. This is called as a booster response. Now, what are the differences between the primary and the secondary immune response? The primary immune response occurs as a result of primary contact with an antigen, whereas secondary immune response occurs as a result of second and subsequent contacts with the same antigen. Antibody level reaches peak in 7 to 10 days in primary immune response, whereas in secondary immune response, the antibody level reaches peak in 3 to 5 days. Prolonged period is required to establish immunity in primary immune response, whereas in secondary immune response, it establishes immunity in a short period of time. In primary immune response, there is rapid decline in the antibody level, whereas in secondary immune response, the antibody level remains high for a longer period. In primary immune response, it appears mainly in the lymph nodes and the spleen, whereas secondary immune response appears mainly in the bone marrow, followed by spleen and the lymph nodes. Now, let us move on to the lymphoid organs. What are lymphoid organs? Immune system of an organism consists of several structurally and functionally different organs and tissues that are widely dispersed in the body. The organs involved in the origin, maturation and proliferation of lymphocytes are called lymphoid organs. Based on their functions, they are classified into primary or central lymphoid organs and secondary or peripheral lymphoid organs. The primary lymphoid organs provide appropriate environment for lymphocytic maturation. The secondary lymphoid organs trap antigens and make it available for mature lymphocytes, which can effectively fight against these antigens. Here in this image, you can see the lymphoid organs in the human body. On the left side of the image, you can see the primary lymphoid organs, the thymus and the bone marrow. On the right side, the secondary lymphoid organs, the lymph nodes that are present in the different parts of the body, the spleen, the Peyer's patches in the small intestine. Now let us move on to the primary lymphoid organs. The bursa of Fabricius of birds, bone marrow and thymus gland of mammals constitute the primary lymphoid organs involved in the production and early selection of lymphocytes. These lymphocytes become dedicated to a particular antigenic specificity. Only when the lymphocytes mature in the primary lymphoid organs, they become immunocompetent cells. In mammals, B cell maturation occurs in the bone marrow and T cells maturation occurs in the thymus. Here in this image, you can see a bird having the bursa of Fabricius. It is a primary lymphoid organ of birds. It is attached to the dorsal side of the cloaca. B lymphocytes mature in the bursa and bring about humoral immunity in the birds. Now thymus. Thymus is a flat and bilobed organ located behind the sternum, above the heart, below the trachea. Each lobe of the thymus contains numerous lobules separated from each other by connective tissue called the septa. Here in this image, you can see the left side shows the thymus, which has two lobes present just in between the lungs, behind the sternum, below the trachea and above the heart. On the right side image, you can see the thymus having lobes separated into lobules by large number of septae. Each lobule is differentiated into two compartments. The outer compartment or the outer cortex is densely packed with immature T cells called the thymocytes, whereas the inner compartment or the medulla is sparsely populated with mature thymocytes. One of its main secretions is the hormone thymosin. Here in this image, you can see the structure of the thymus gland. It has a capsule. Inside the capsule is the cortex, and the cortex is separated by large number of interlobular septum. Inside the cortex is the medulla, which has large number of thymic corpuscles. It stimulates the T cell to become mature and immunocompetent. By the early teens, the thymus begins to atrophy and is replaced by adipose tissue. Thus, thymus is most active during the neonatal and the pre-adolescent period. After the pre-adolescent period or after the adolescent period, the thymus shrinks. Bone marrow is a lymphoid tissue found within the spongy portion of the bone. Bone marrow contains stem cells called the hematopoietic cells. These cells have the potential to multiply through cell division and either remain as stem cells or differentiate and mature into different kinds of blood cells. The process of production of blood cells in the bone marrow is called hematopoiesis. Here in this image, you can see a portion of the iliac bone showing the outermost periosteum, below which is the compact bone. Below the compact bone is the spongy bone. In the center of the spongy bone is the bone marrow, which is responsible for hematopoiesis. Secondary or peripheral lymphoid organs. In secondary or peripheral lymphoid organs, Antigen is localized so that it can be effectively exposed to mature lymphocytes. The best examples are lymph nodes, appendix, Peyer's patches of gastrointestinal tract, tonsils and adenoids, spleen, MALT, mucosal associated lymphoid tissue, GALT, gut associated lymphoid tissue, BALT, bronchial or tracheal associated lymphoid tissue. Now let us move on to the lymph node. Lymph node is a small bean-shaped structure and is part of the body's immune system. It is the first one to encounter the antigen that enters the tissue spaces. Lymph nodes filter and trap substances that travel through the lymphatic fluid. They are packed tightly with white blood cells, namely lymphocytes and macrophages. There are hundreds of lymph nodes found throughout the body. They are connected to one another by lymphatic vessels. What is lymph? Lymph is a clear, transparent, colorless, mobile and extracellular fluid, connective tissue. As the lymph percolates through the lymph node, the particulate antigen brought in by the lymph will be trapped by the phagocytic cells, follicular cells and the interdigitating dendritic cells. Lymph node has three zones: cortex, paracortex and medulla. The outermost layer of the lymph node is called the cortex. It consists of B lymphocytes, macrophages and follicular dendritic cells. The layer inside the cortex is called the paracortex, which is richly populated by T lymphocytes and the interdigitating dendritic cells. The innermost zone is called the medulla, which is sparsely populated by lymphocytes, but many of them are plasma cells which actively secrete the antibody molecule. Here you can see the cut section of the structure of the lymph node, where it has an outermost capsule. Inner to the capsule is the cortex. The cortex consists of large number of sinuses and the mantle zone containing the follicles. Inner to the cortex is the paracortex. Inner to the paracortex is the medulla, which has large number of medullary cords. You can see that the afferent vessels entering into the lymph nodes and the efferent lymphatic vessels leaving the lymph nodes. As the lymph enters, it slowly percolates through the cortex, paracortex and medulla, giving sufficient chance for the phagocytic cells and dendritic cells to trap the antigen brought by the lymph. The lymph leaving a node carries enriched antibodies secreted by the medullary plasma cells against the antigens that enter the lymph node. Sometimes visible swelling of the lymph nodes occur due to the active immune response and increased concentration of lymphocytes. Thus, swollen lymph nodes may signal an infection. There are several groups of lymph nodes. The most frequently enlarged lymph nodes are found in the neck, under the chin and in the armpits and in the groin. Here in this image you can see the different groups of lymph nodes in the body: the cervical lymph nodes, the supraclavicular lymph nodes over the clavicle, the axillary lymph nodes in the axilla, the mesenteric lymph nodes in the stomach, the iliac lymph nodes, the inguinal lymph nodes, the popliteal lymph nodes, the lumbar lymph nodes. All these lymph nodes drain into from the lower part to the cisterna chyli, and through it is carried through the thoracic duct. What is cisterna chyli? It is a dilated collecting sac located in the abdomen in front of the bodies of the first and the second lumbar vertebra. It drains the lymphatic trunks of the lower part of the body, give rise to the thoracic duct, which ascends through the aortic opening of the diaphragm into the thorax. Now, what are lymphatic ducts? The cisterna chyli is the most inferior part of the thoracic duct. The thoracic duct empties into the junction of the left internal jugular and the left subclavian veins. The right lymphatic duct empties at the junction of the right internal jugular vein and the right subclavian vein. What are Peyer's patches? They are oval-shaped areas of thickened tissue that are embedded in the mucus-secreting lining of the small intestine of the humans and other vertebrate animals. Peyer's patches contain a variety of immune cells, including macrophages, dendritic cells, T cells and B cells. Here in this image, you can see the intestinal lumen, the microvilli that are present. Below the epithelium of the microvilli is the lamina propria, which contains the M cells and some follicles. Below the lamina propria is a layer of submucosa, which is filled with multiple follicles having the germinal centers. The follicles are together called gastrointestinal tract Peyer's patches. The tonsils, otherwise the palatine tonsils, are a pair of soft tissue masses located at the back of the throat or pharynx that is present between the anterior and the posterior fosses. The tonsils are part of the lymphatic system, which helps to fight against infections. They stop the invading germs including bacteria and viruses. Here in this image you can see the location of the tonsil, which is present between the anterior and the posterior fosses. The center you can see the uvula, above it is the soft palate and the hard palate. The adenoids, they are glands located on the roof of the mouth, behind the soft palate where the nose connects to the throat. The adenoids produce antibodies that help to fight infections. Typically, the adenoids shrink during adolescence and may disappear by adulthood. Here in this image you can see the location of the adenoids just behind the mouth, between the junction of the nasopharynx and the oropharynx. Spleen, it is a secondary lymphoid organ located in the left side of the upper part of the abdominal cavity close to the diaphragm. Spleen contains B anti-lymphocytic cells. It brings humoral and cell-mediated immunity. Here you can see the anatomy of the spleen, the location of the spleen on the left side image, and the right side image you can see that the cut section, it has a capsule surrounded by a red pulp and the white pulp in the center, and these are separated by a large number of trabeculae. Mucosa associated lymphoid tissue, otherwise called MALT. MALT is a diffuse system of small concentrations of lymphoid tissue that are present in the alimentary tract, respiratory tract and the urogenital tracts. MALT is populated by lymphocytes such as T and B cells, as well as plasma cells and macrophages, each of which is well situated to encounter antigens passing through the mucosal epithelium. It also possesses IgA antibodies. Here in this image you can see the mucosa associated lymphoid tissue. When any foreign antigen like the bacterium enters into the cell, it is devoured by the macrophages, which stimulate presents the antigen to the T cells. The T cells in turn results in the stimulation of the B cells, forming the plasma cells, and the plasma cells release the IgA. The IgA gets attached to the receptors of the mucosal cells, and this IgA is secreted outside into the lumen. These B cells may further produce large number of IgA through the plasma cells, and this IgA can also migrate to other mucosal membrane. Gut associated lymphoid tissue called GALT. GALT is a component of the mucosa associated lymphoid tissue. It is a type of MALT which works in the immune system to protect the body from invasion in the gut. Bronchus associated lymphoid tissue called BALT. BALT, also a component of MALT, is made up of lymphoid tissues in the tonsils, lymph nodes, lymph follicles, is found in the respiratory mucosae from the nasal cavities to the lungs. Here in this image you can see the different types of MALT tissue. The nasal associated lymphoid tissue like the pharyngeal tonsil or adenoids, palatine tonsils, lingual tonsils and the upper respiratory mucosa. BALT, which is associated with the bronchial associated lymphoid tissue, the lower respiratory mucosa and the bronchial patches. SALT, which is associated with the skin. GALT, intestinal mucosa, Peyer's patches and the appendix is included in GALT. So today in Human Health and Diseases Part 4, we discussed about the basic concepts of immunology, in which we talked about the types of immunity, immune responses and the different types of lymphoid organs. So thank you. Kindly subscribe, like, share and comment the channel Read MedPrep Academy. Kindly register for UG and PG NEET type MCQs in our website www.readmedprepacademy.com. Our Facebook ID is Read MedPrep Academy. Our email is readmedprepacademy@gmail.com. Our Instagram is Read MedPrep Academy. Kindly join Read MedPrep Academy and WhatsApp the number given below. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.